But in terms of really like bigger environments, the soil, the ocean, the water, the human gut, I think we need to move beyond knowing who is there to knowing what they are doing and what the products.
Something to Chew On is a podcast devoted to the exploration and discussion of global food systems. It's produced by the Office of Research Development at Kansas State University. I'm Maureen Olevnik, Coordinator of Global Food Systems. I would like to welcome co-host Dr. Mark Crosby. Dr. Crosby is Associate Professor in the Department of English and is the Director of the K-State Digital Humanities Center. And co-host Dr. Jim Stack. Dr. Stack is Professor in the Department of Plant Pathology and Director of the Great Plains Diagnostic Network. Study of the microbiome is diverse and complex. From its start 50 years ago to today's efforts, we have moved from evaluating single organisms to the realization that the study of multiple organisms, their interaction and functional characteristics is required to begin understanding the microbiome. These evaluations encompass all forms of life, including the human and animal gut, living soil, plants, and sea life. We are clearly in the infancy of understanding how those organisms impact our health, a plant's ability to grow, and literally everything that deals with life as we know it. In today's podcast, we talk with Dr. Sonny Lee, Associate Professor in the Division of Biology at K-State. Dr. Lee's work touches on many areas of the microbiome research and casts a wide net in laboratory studies, mining data from peers to aid in complex evaluations, and working with students and colleagues capable of critical thinking and creative problem solving. I'd like to welcome Dr. Sonny Lee to the podcast today. Dr. Lee, could you give us a little background on who you are, how you came to study and enjoy the work that you're doing, and how you came to K-State?
Who I am is a very difficult question to answer. The reason why this is so difficult is because this is my second career. My first career, in fact, a lot of people do not know, is I was a film director for a long time. And then I became a scientist, all because I wanted to study coral reefs, all because I love diving. And it took me all the way from Florida to New Zealand, where I started understanding how important the microbes is to the environment, to the coral reefs. And after I got my PhD in New Zealand, I decided that I like to pursue this direction on microbes, not necessarily just coral reefs. And what I did was just like, I asked myself, and I think that what I'm studying is not enough. I do not want to know who are these microbes, but I also want to know what are these microbes doing. And I started going online looking for tools to answer this question, and I stumbled upon my ex-PI or my postdoc PI's work. And I started using his software, and that was the time I spotted an error. I sent him an email and said, hey, there's some error with your code, your program. And he sent me a reply and said, who are you? And I said, well, this is me. And before I know it, I was in Chicago, University of Chicago, and started working on a postdoc with him. And the next thing I know is I started to understand the limitations of all this work with bioinformatics. And I wanted to take one more step and to understand mechanistic on how this set of microbes do what they do and not just what they are doing. And then I proceed on to do one more year postdoc with another PI who is purely on mechanistic and have no idea about sequencing. So I spent 1 year teaching him about sequencing while getting all the things I know from him in terms of mechanistics. So after about 3 years in University of Chicago, I arrived in K-State in August 2019. And since then, I've been working on host micro interaction on a whole bunch of different model organisms, from plants, big blue stem grass to the pigs to human and as well as mice model.
That is an interesting background. It's not one of those lines that you draw straight from one portion to another. So that's quite interesting. I'm going to, just reading through some of the work that you've been doing, I'm going to ask a very basic question because I would like anybody that listens to this to understand basically what we're talking about. When I'm reading through, you're talking about genomics and you mentioned it a few times. Then I read through and you work on metagenomics, metatranscriptomics, metabolomics. What is the meta piece of that? The genomics I understand. What is the meta piece and how does that work together in those different areas of study?
So when we talk about meta means everything. And in a lot of sense, when we talk about genomics, at least in the field that I'm studying, we are talking about one single organism, the genome or the genomic information of one single organism, one single bacteria, one single model organism. But when we talk about metagenomics or metatranscriptomics or metabolomics, every single thing that encompasses within the item. So for example, if we are talking about metagenomics of a pig or a plant, we are talking about every single gene within that plant or within that pig. Every single gene could be encompassing everything from bacteria to the fungi to virus to even the pig itself. Every single gene, we are studying everything in there, and we are not just looking at one single portion of it.
Hi, Sonny. Can I take you back to your first career as a director?
Yeah.
Like you, I've enjoyed diving in the Caribbean and in Australia, the Great Barrier Reef. And I just enjoyed looking at the beautiful things down there and getting slightly nervous when a hammerhead shark would swim by. But could you go into more detail and explain what exactly is going on with bacteria and coral reefs? And then how did that inspire you to go on to your present career?
What happened to the bacteria, or how do they interact with the coral host by itself? In short, nobody knows exactly, right? If we know, there's nothing to study anymore. The coral reef itself is actually a very complex organism. The coral reef is multiple coral reefs living together. So if you think about it, each single polyp that is like an octopus inverted is one coral. And in the structure you see underwater is making out of millions and millions of corals, all living together, packed together like in a big apartment. And within the coral itself, there is a very distinct symbiome, and this is algae. And the algae is a different type of algae that live with the coral. And then within this coral itself and the algae, between them, they have these microbes. And it could be bacteria, and it could be virus, it could be other things. And bacteria, of course, makes up almost 80% to 90% of all the microbes in there. And what do? A lot of people are speculating that they help with nitrogen fixings because this coral lives in a very, very clean water, which in fact, the nutrients is really low. So they get some of the energy from the sunlight, which the fungi within the coral fix it and give the food to the corals. But at the same time, some of the nutrients are fixed by the bacteria and give it to the coral as well. And in return, what do the bacteria and the fungi get in return is they get a place where they can be safe, because these coral reefs are really built of skeletons, right? Carbonate skeletons where they can be not just swimming around in the ocean and get eaten up by other things. So that's one thing, is providing nutrients. And second thing is in terms of climate change, there has been quite a fair bit of hypotheses or speculations, is that some of the interactions between the coral itself and the fungi, as well as the bacteria, helps to make this coral host a bit more resilient when there's a change in temperature, or when there's a change in acidification or other parts of it. This is a really difficult field to study, and a lot of researchers are working on it, and they are really good researchers, obviously. And we probably will not know in the next 5 to 10 years. But as we get more and more information, this knowledge gets clearer and clearer what the microbes is doing with the fungi as well as together with the host itself.
Thanks, Sonny. So one of the things I'm hearing you say is that with these coral reefs, you get sort of multiple organic systems, right, that exist in an interdependent or reciprocal relationship that can adapt or evolve depending on, as you mentioned, changes in climate. One of the things I noticed actually when I was diving in the Caribbean, that there's a lot of coral bleaching, which suggests that reciprocal or interdependent relationship becomes sort of unbalanced. And I guess what you were talking about with the research, analyzing, why these imbalances, what are the causes of these imbalances, and perhaps how we can address them.
Yes, a lot of research being pointed to the direction of trying to understand these mutualistic or mechanistic interactions between the coral host, and they call it coral holobiont, which includes everything within the coral host itself and including the fungi. For a while, we started to understand that bleaching of the coral is a result of the breaking down of this relationship between the fungi and the coral host itself, where what is happening is, for a long time, the hypothesis was that the coral host could no longer host the fungi, and as a result, the fungi either leave the host or they're being expelled out by the host. And there are quite a fair bit of hypotheses and rationale on that, but none of them has been really proven at this present moment. And now we have the other bacteria that comes into play, and so right now there's this three-way interaction, and a lot of studies are trying to understand how this three-way interaction works. Again, I emphasize that this is rather difficult because if you think about it, comparing a coral to, say, a human, all our microbes are most of our microbes, let's say the guts are inside. And if you think about a coral is literally the gut that has been flipped inside out, where it's now exposed to the environment. So every single research, any single work has to take into account that whole big ocean that is swimming around the coral itself suddenly just get 10 times harder.
Yeah, great. It's as if on a sort of oceanic level, the gut has been extruded. I think that's what starfish do, don't they? They extrude their guts to digest food. As you brought up, can you talk more then about this transition from looking at that relationship between the fungi and the bacteria to your current research, looking at the microbiome? And I wonder, for someone like me who's a humanist, could you start with the definition of a microbiome?
Yes. So in my context, when we talk about microbiome, is everything within the host that we could not visualize by eye. It could be bacteria, which took a very big percentage of it, virus, fungi, protists, and of course, bacteria is one of the biggest items we study in our lab because it encompasses at least, if more 80% of the whole microbiome. So the transition or the addition of the human microbiome to the coral reef research happens at University of Chicago, or it begins at the University of Chicago, where my postdoc PI during then was interested in ocean, but they are more interested in the human aspect of it as well. And one of the big items that we were studying, and right now my lab is studying, is inflammatory bowel disease in patients. So inflammatory bowel disease is quite prevalent in US and the world. And these patients actually, or these individuals actually doesn't really have a really good lifestyle because they get a lot of inflammation in their gut. And we wanted to understand how and why it happens, and is there a way to prevent it? And in terms of inflammation bowel disease, or we call it IBD for short, comes in kind of three components. The host components, which is like the genetics of the host. If your parents have IBD, there's a high chance that their children will have it. And then there's the environmental component. It depends on what you eat on a daily basis. If you eat lots of McDonald's from day 1 to day 10 every single day, there might be a bit of chance that you will not really have a good gut microbiome. And the third thing, of course, is how the microbes interact with the host, causing all this inflammation in the gut. And our lab more interested is in the microbes and how the hosts interact with each other. So that is where we are at the present moment. We have been using mice as a model to investigate how does antibiotics result in the IBD development in later stage of their life. And we also investigate how does the host genetic factors comes into play in this interaction, and is there any way we can intervene, cause an intervention with these microbes at the very early stage so that the IBD can be prevented at the latest part of their life.
Yeah, if I could maybe follow up on that a little bit. One of the challenges that I think are common to a lot of areas of study is dealing with variation. So you use the example of if you eat Big Macs for 10 straight days, it may very well alter the gut flora. So when you look at the different systems you've studied, coral systems, prairie systems, human biome systems, how do you deal with variation? Meaning that how many people do you need to sample over what time frame to get a sense of what normal is versus what is transient because of the diet or just their stage of development, things like that. I mean, you look at a prairie and maybe there's a copse of trees in one place and there's a stream in another. And so you wouldn't, and we know from sampling that it's not uniform. How do you get a sense for what represents what you're studying?
The answer to that, I'm always saying, is as much as I can afford, or as much as anyone can afford. And that's obviously the truth. I think one of the advantage of using metagenomics or metatranscriptomics to study a disease or a microbe evolving phenomena or phenotype is the amount of data that has been generated over years and years by other researchers. What do I mean by that? I can give you an example. So I'm going to kind of step away from human microbiomes to plant, but it is exactly the same way. So for example, in our lab, what we are doing is we are looking at how does microbe contributes to the resilience of the plant host in terms of drought. And we conducted drought-induced experiments on big blue stem in the greenhouse. And of course, in anyone who conducted greenhouse experiments knows that it may not be entirely the same as in the field. And we acknowledge that as well, and we know that. So when we did an experiment, we came up with a group of microbes that we are interested in. And we assembled their genomes. And right now, for example, we have these 10 genomes, 10 bacterial genomes that we assemble from the greenhouse. And the next question we will ask ourselves is that, well, does these 10 genomes that we assemble even make sense in the real world, in the prairies out there, in the grassland, in the forest? Or is what we are thinking about only in the greenhouse and it doesn't make sense at all? So what we did is we verified these 10 bacterial genomes. And we went to all the published data as much as we can that has been published, whether on the database or public database. We download all these metagenomes from all the studies. And right now we have all these samples that we did not study, but somebody else has studied over years and years, right? And we have all their genomic sequences. And what we did is we tried to find our 10 bacterial genomes among all these samples that has been published over years. In a lot of ways, you will then be able to see the pattern of these 10 genomes start showing up in the prairies, in the grasslands, in the forests, in the mountains, or any plants. How much are they detected? Are they only detected in grasslands, or are they detected only in forests? Are they detected during the drier months or during the winter or in the wetter months, or where are they located exactly? And this gives us a glimpse of, one, is the bacteria we assemble really prevalent throughout the other locations? And two, when they show up in other locations, do they still maintain the same genomic information, or do they change what we call in terms of strand level? And when they change, what kind of functional change would we expect? Say, for example, when we grew these bacteria, we found them in the greenhouse, and then we saw them in the prairie in March. Do they look exactly the same, or in terms of really on the genomic level what has changed. In short, is we cannot do as many experiments as we like, or as many we call replications as we like, but what we have is we have other publications that have been done, and we use them as our replicate.
Has anything surprised you and said, what is this doing here?
I'm going to shift again from plants to the swine. And one of the things was interesting about the swine microbiome, or at least the swine gut microbiome, that over the last 2.5 years was trying to make sense of it, was we studied this swine from very young pig, pre-weaned pig to adult. And we found a very distinct set of archaea, and they are not bacteria throughout the whole life. And this archaea kind of goes up and down in terms of abundance. And we were wondering why. And so about 2 months ago, where we took another step, was we took this archaea genomic information, and we started downloading all the other samples from other studies, and we mapped these archaeas to other samples. And we were trying to see if they even show up, but the more interesting thing is that they show up in terms of exact pattern that we have done, even though they are from other locations and other studies. Yeah. So that was something that we were just like, oh, wow, this is really unique. And right now we are trying to go one more step further and looking at culturing this archaea. The reason is this is because it's like, okay, right now we know that this archaea shows up in pre-weaning and nursery pigs, the younger pigs, as much, and not so much in the adult pigs. In the adult pigs is another genus of archaea show up. So we say, okay, let's culture the one that is a young pig's archaea. And what we like to do is then we challenge it with the bacteria and see if they actually do anything in these interactions. Because in the gut environment, the archaea doesn't live alone. They have to live together with the bacteria. And are they doing anything that causes it to switch over to another another species of archaea in the later stage of the life? So that's the part that we are working on right now.
Yeah, pretty interesting.
Sonny, can I take you back to your description of how the microbiome affects sort of inflammatory diseases? I wondered if you could sort of explain more about how do microbes inform or influence inflammation?
I guess, again, the short answer to this is we do not know. I feel really great when I say we do not know, because it means that I'm still in the business. So there is a lot of aspects, and we always thought that some of the bacteria may be pathogenic, but we do not know enough. And as far as we know, or as far as I know, there might be just only a few bacteria that's potentially pathogenic. And in a lot of sense, I think is that certain bacteria interact with the host in a way that affects the host's immune response that causes the inflammation. For example, this is one of the aspects the lab, one of the graduate students is looking for her PhD work. Let me take a step back. When a mom is pregnant and when they take antibiotics, these antibiotics kind of wipe out some parts of their gut microbiome. But what's more interesting is that this gut microbiome from the mom is actually passed down to their offsprings. So we use a mice model to test that and demonstrate that. We feed antibiotics, broad-based antibiotics to the dams during their pregnancy. And when you examine the gut microbiome of their offsprings, the pups, you would see that they are missing certain microbes, which is also not existing in the dams, which is like, oh, wow. So microbes are actually transmittable generation-wise. But then what's more interesting is that as these mice, they get older and older, some bacteria shows up at the later stage of their life. And when this bacteria shows up, it coincides with the timing of the gut inflammation. And this let us kind of believe or kind of guess that maybe what is happening is this bacteria already exist in the gut, but they are very low in abundance in the early stage. And because they are low in abundance, they do not educate the immune systems of the host. So when these microbes show up at the later stage of the host, of the life of the host, the immune system of the host just literally just freak out and launch a full attack and results in inflammation. So we say, well, if that's the case, can we introduce these microbes to the host at a very early stage and see if they show up later back, they will be all right. And we did a lot of tests. We introduced different microbes. We introduced all the microbes, and we introduced at different stage from 3 weeks all the way to 11 weeks. And I think we're starting to get some results back on the introduction. And some of the results looked pretty promising, where if the microbes are introduced really early in the pups or the offspring's life, as early as 3 weeks of age, they actually have very lesser inflammation as compared to those pups that have no introduction of microbes in the early stage of their life. So this is something that we got really interested in and started to get further into a mechanistic and say, okay, these microbes that we introduce, what are the microbes that could be causing this impact? Our lab and also a few labs that we know pretty well and we start to work together was all coming together on this one single E. coli. And we have, for a long time, we start wondering, so what is E. coli doing in here? Because E. coli has hundreds of strains and from commensal to pathogenic. And so we have been trying to identify what is this E. coli and what is it doing and what kind of pathways results in this E. coli being the bacteria that all of us are so interested in so far.
Thanks, Sonny. One of the things I picked up from your previous discussion was that sometimes there are triggers or environmental triggers for these dormant microbes that kick them into gear. And you used the example of eating too many Big Macs. And it reminded me of the "you are what you eat" somewhat cliche adage. How far in your research would you say "you are what you eat" is an adage, even if it may be cliched?
I think that is very true. For example, long time ago, I was playing my dog and it bit me on somewhere on my left thumb. And for a while, I was just like, well, it was just a scratch. It doesn't matter. And then 2 days later, it started turning brown. I said, well, it doesn't matter. It's just a bit of infection. I'll just do something on it. It's fine. And then 3 days later, it starts swelling. And I start drawing lines on it because I know if we start traveling, it means bad news. And before you know it, I said, oh no, I probably should go and see the doctor right now. And I went to ER and they were like, yeah, the best patient we ever had. You track every single day where is the movement of the bacteria. Well, the thing is, they gave me broad-based antibiotics. And knowing what I'm knowing, I studied that very well. And I know that this is going to screw me up quite badly with these antibiotics, broad-based. And I was on broad-based antibiotics for nearly a week. And I could feel my motion or my poop wasn't as normal as what I used to be. And lucky for me, I guess, is I know what I will probably, the antibiotics is wiping what type of bacteria away. And I also know what I should be eating to try to regain the whole balance. And I ate a lot of green vegetables almost for about a month and a half to 2 months. And I avoid a lot of dairy products. I avoid a lot of red meat. And in about 3 months, I was almost back to normal. I guess I'm lucky in that sense, but I think what we eat is definitely shows up at the end of the day. I think genetic plays a very strong factor as well. But beyond genetics, the next level is definitely the diet or definitely what we eat as well. Yeah.
So I know you just touched upon that. Could you say a few things about what we should be eating and perhaps what we should be avoiding?
Yeah, I think there's a book that I read from Microbiome about diet, and I think he has kind of nailed it because I'm not a person who believes that we should be eating one type of food and not the other type of food. I believe that we should be eating what we love to eat. But from Microbiome's book, I start to realize that, well, I do not eat anything, at least for the last few years, anything that is within those aisles, which means all those canned foods. I try to get food that is on the outside of the grocery store, which means that all the vegetables, all the fresh meats, all the fresh fish and stuff. And I also follow what he said, very not strictly, but almost to the sense of 80-20. I mean, 80% of the times we eat well. That means we eat lots of vegetables, lots of salad, and 20% of the time I cheat. I go to McDonald's, I go to Burger King and Taco Bells and stuff like that. And I think it is a good, well balance of life that makes us good.
I'm going to bring this back to the agricultural side of things because a lot of what you do touches right into that area. Last podcast we did was with Dr. Kumar, who is a weed specialist, works on weed control in production. Have you ever looked at, or do you have a feel for something like the impact of pesticides on the microbiome in the soil? So we do a lot of things in the agricultural community that directly impact what's in there. Have you looked at that, or do you have a feel for that?
I am on the outside committee of this student who is from Africa, and she is very, very hardworking. And she is conducting the impact of herbicides and pesticides on the soil microbiome. And she is at this present moment analyzing her data, but briefly is that there is a big change in terms of the soil microbiome after spraying with herbicides or pesticides. And we do not know yet is how long does this change or this impact on the soil microbes. Are they going to persist just a year, 2 years, or how long does they persist? We do not have that data yet. One of the things that during conversations between her and I in the very beginning of her thesis work, beginning of the research, was a tool that I use in other research, whether is it human or plant, is I use what we call fecal microbial transplantation. Right? We transplant something to a healthy microbes from a healthy individual to a diseased individual, from a healthy plant to a non-healthy plant, from a plant that it can resist drought to a plant that cannot resist drought. And I asked her, can we do the same thing or transplant some of the microbes from a healthy soil to a non-healthy soil? Do you think they will kind of persist and they will hang on there or not? And we do not know. We still kind of wondering about all those questions.
You know, to kind of follow up on Maureen's question, I think there's enough evidence out right now that studying the microbiome is pretty important. So we need to, as you suggested earlier, Sonny, we need to keep diving deeper into this to understand. But we're also at an important time in Earth's history with respect to the population and how much we're impacting the Earth that we live on. If we want to really assess the impact of some of these major things like climate change, we need good data sets. Have you thought about this? Do you have any recommendations for what data we should be collecting in environmental systems, agroecosystems, or even forest ecosystems? What data should we be collecting about the microbiomes that will help us evaluate what these impacts really were, say, 30 years down the road?
I do have thoughts, but I cannot say that is definitely correct. The microbiome study came, 50 years ago, where we started to understand maybe how important the microbes are into the environment. And during that point, our technology is not really catching up that fast, and we study microbes based on who they are. And then in the last 15, 20 years, we started to get a sense that, well, two microbes, they might be doing different functions, even though they may be the same genus or maybe the same species. Otherwise, two microbes from the same genus may be doing different functions. So we started to need to understand what is their function. And personally, I think that we are at a stage where we have more information than we can handle per study. And every single study nowadays are really like interdiscipline. They will have, in terms of climate change, they will have people who study the soil, study the environment, study the microbes, study the plants, the genotype, everything. And of course, we would like to collect as much information as possible. And then in terms of the microbes, what should we collect is we should try to move beyond knowing who is there, but thinking about what they are really doing, and even to the extent of what products do they produce. And that's why we come out about the metabolites. And unfortunately for us is that even though we are moving so fast with technology, the cost itself is decreasing, but not decreasing fast enough for our grants to catch up. So for example, in studying who is there, we usually use a marker genes. And marker genes used to be $200 per sample. And right now, after so many years, it could be as low as $10 per sample. Whereas in terms of looking at what kind of functions they may be doing, right now is about $150 per sample. And it depends on a lot of factors. Example, in the soil community, soil microbial community, is very complex. So you need to sequence what we call sequence deeper, which means that per sample, instead of just sequence one time on the machine, it could be sequencing like 5 times on the machine. And that means you are looking at 5 times the cost. So at the end of the day, it is an important balance of like how much we have to spend on certain things versus how much information we want to get from there. And at this present moment, my personal opinion is that just knowing who is there, what microbes are there, may no longer be something that is giving us more insights than we have done for the last 50 years. Unless this is a very unique environment, such as the deep water thermal vents, then we may not necessarily need to know exactly what they're doing because they are just a very small set of microbes. But in terms of really like bigger environments, the soil, the ocean, the water, the human gut, I think we need to move beyond knowing who is there to knowing what they are doing and what the products. And what we have to do probably is we have to then take a step back and say that, well, if we cannot get as many replicates, maybe what we have to do is get less replicates, but more high resolution, more in-depth. And then with the in-depth, with the more details, we then use other database or other publications that have done before us to see a wider aspect of it.
Just based on what you just said, one of the questions that I had marked down was referencing your noting of the diversity of teams that you have put together. And I think I saw you had like a job posting saying that you aren't necessarily looking for somebody that's in deep understanding of what exactly you're doing, but may have a broader curiosity and involvement in other areas of research. What are your thoughts on that interdisciplinary approach to things, on the need for the diversity in tackling problems like this and the kinds of specialties that you see being brought into studying these types of things?
In terms of interdiscipline, I think our research in general, why it's not just my research, is moving into the direction. I think gone are the days where we are just doing one single thing by ourselves in our garage and don't worry about everyone else, what they are doing. And right now, what we are doing is going to be pulling together in every single direction. For example, in our lab, I do not believe studying one single microbes can give a lot of insights to a community, which is the microbiome. I also do not believe studying the microbiome as a whole can give you any insight into the mechanism. And so I'm kind of in between. What we came up with this idea, and we have been following this for the last 2 years, we came up with what we call a simple state community. Instead of looking at the whole microbiome or looking at one, why can't we look at a functional group that probably drive the whole microbiome somewhere? We were looking at one single function that we're interested in, then we drive the microbiome to that place, and then we look at the mechanistic within much more simple microbes. Then I was looking at my lab trainees and looking at myself and say, well, I do not know what this molecule is doing. We need a biochemist. Well, I do not know how these microbes is doing all this stuff in the soil, and we need someone a geologist to tell us all the biochemical pathway that's going in the soil. And I do not know what is these microbes interacting with this plant, and how does this interaction drive the plant, or how does the plant drive the microbes. We need a person who knows about the plant's genomes to talk to us. So, and in short, I'm starting to pull all these people in together, and we can work together and get a better sense of how this whole, I guess I don't call it just microbiome, I call it a holobiome, HOLOBIOME, work together. Because if I'm just working on the microbiome and just ignore everything else, it's just same thing, I'm just working in my garage again. And I like to start to understand how the whole thing as one single entity. And I think that is the most important part for me. And bringing in different expertise is the best way to move forward in my research in the scientific community.
Sonny, when you talk about focusing on the function, is that where the metatranscriptomics comes in?
Yes. So the study of what our microbes, we have different levels. And the very start is the marker genes, who are they? And then we go on to the metagenomes. In metagenomes is who are they and what could they be doing, which means potentially what could they do. And then the metatranscripts, who are they and what they are exactly doing. Because you have the potential, that doesn't mean that you will be doing that unless maybe it's in the drought conditions. And then after that is the metaproteins, right? What proteins is being produced by this function. And then we have the metabolomics, is what kind of metabolites that is resulting from all this interaction. So these are the different levels. At the present current situation, our lab and a lot of other labs has no problem linking from marker genes to metatranscripts. There's always a very difficult or a challenge to try to link the metatranscript to the metaproteins and the metabolomics, because the proteins that is produced or the metabolites produced could be coming up from a lot of different pathways. And that bridge is currently the challenge of the bioinformatics or the study of the microbiome in terms of crossing over that challenge hurdle.
How do students prepare to contribute to this area? What do they need in their education that would help tackle some of these questions?
I think one of the most important thing, critical thinking and creative thinking. Bioinformatics is hard to a lot of people, but it's not the most crucial thing, because in very few circumstances, we do not develop programs. We use programs that has been in existence, and we find the biology behind the questions using these tools. And in a lot of sense, most of, if not all the challenges or problems, issues you face with bioinformatics, someone else probably had done it and faced the same problem. And quick Google it, you will see, oh, this is a solution. And you can adapt that solution to your challenge, your issue. Managing big data is challenging because there's so much information out there. But finding that biology, finding that function, finding that molecules that is among this huge data, that is the most difficult part, which none of the researchers, none of the scientists can teach any student. It is up to the students to go through this data and say, well, this is an interesting function, and it relates to this biology, and it relates to this environment, it relates to these parameters. Maybe this is what is happening. And that is the part which needs the students' creative and critical thinking. And that is the part which I don't think any scientist or any PI or any supervisor can teach the student. And I think that's the part which any student should start developing right now.
Yeah, thank you. Yeah.
It's heartening, Sonny, to hear you say that you encourage students to think critically and creatively, because that's what we try and do over here in the humanities. This is the goal that sort of drives us, that they should think critically and creatively about whatever text or image that they're encountering. Can I go back to your discussion on the importance of interdisciplinarity? And could I invite you to talk a little bit about your work with the Johnson Cancer Research Center and what you're doing there at the moment?
Yes, with the Johnson Cancer Center, specifically, we are working on the IBD model, which almost results in colon cancer in a lot of circumstances. At the present moment, our lab have 3 cancer models, and IBD is one of them. And there's 2 other cancer models that are in development, which we hope to work with the Johnson Cancer Center in very near future. One of them is a head and neck cancer, which I work with a collaborator in KU Med very closely. And we had a really preliminary study with 7 patients that have head and neck cancer. And we took their saliva and we compared them to their spouse who were healthy. And well, we found that the microbiome obviously first is different. And then we started to look in depth into the function and the metabolites. And there are metabolites which are way different, and they also hypothesized to be carcinogenic. And those are the things that we're like, oh wow, this is really cool, let's start looking some more. And so that's one of the cancer model. And the other cancer model, again, is I'm working a lot with KU Med, is a radiation model, which we know that we subjected these mice with radiation, and we found that not only the microbiome is different, and this mice doesn't really survive radiation, post-radiation very well. But what happened is that we kind of do a reverse engineering, and we gave some of these mice some of the microbes which we hypothesized to be relieving this post-radiation stress. And these mice kind of live slightly longer than the rest. And right now we are trying to investigate what is going on here and stuff like that. I think in terms of Johnson Cancer Center, we work with them on the IBD model, but I think in the near future, I'm hoping to work with them a lot more in other models as well. And I talked to Johnson Cancer Center Director Dr. Sherry Fleming a couple of times. And one of the few things that I really hope to bring across to Johnson Cancer Center is a collaboration with the medical center. I have been very lucky that I work with the KC Medical Center a lot because of my work. And I hoping medical center and the Johnson Cancer Center and our basic research at the biology or on the college level or on the university level to come together and we can work together, give more insights to what we can better things for the scientific community.
Thanks, Sonny.
Are there any last questions from Jim or Mark?
So, Sonny, you are involved in a lot of things. I'm wondering, do you sleep?
Yeah, that's what my students ask me every day. I think fortunately or unfortunately, I only sleep like 4 or 5 hours a day. I don't really sleep much, but I think that reflects on my pale complexion. That's what I do. Yeah.
I very much enjoyed the discussion, Sonny. Thank you.
That's great.
Thank you, Jim. Thank you, Mark.
Yeah, Sonny, do you have any questions or comments?
No, I don't have anything else. Thanks, Maureen.
Thank you so much. If you have any questions or comments you would like to share, check out our website at ksu.edu/research/global food and drop us an email. Our music was adapted from Wayne Gowen's album, Chronicles of Carmela. Special thanks to him for providing that to us. Something to Chew On is produced by the Office of Research Development at Kansas State University.